everyone. My name is Elna Gottlieb, and I'm an instructor from the DNA Learning Center. And we're going to do some exciting stuff today. So we are actually going to learn a little bit about one of the most fascinating molecules on our planet right here. And so we're going to talk a lot about what this is. And so now, on the other hand, also, not only are we going to learn a little bit about this, but you're going to get to build your own model. So I'm going to show you how to build a model, and we'll see if you have all the things, that's great, because they are things that you sometimes can find around your house. But if you don't, that's all right. A lot of these things are really easy to get, and so you can always refer back to the video at some point. Now, here are some of the things that we are going to need, and I will be doing this near the end of my presentation. But first of all, we're going to need some wire. And the wire, if you don't have wire at all, nothing like it, then that's okay. You can make it without the wire, but it always makes it a little bit better. Now, this is called tag wire, very skinny, but also it's kind of hard to find. And so, you know what? I'm not going to use this wire. No, forget it. Instead, I'm going to use some things that are easier to find. I'm going to use 2 pipe cleaners. And the pipe cleaners, I picked pink for fun. Really, any colors, it doesn't matter what color. In fact, it's going to be mostly covered. So if you want to use your most hated color, that's fine too. And I picked pink just for fun. And so you're going to need 2, and you want to keep them relatively straight. You don't really want them bending. That's going to make it harder to work with. And so you're going to need those 2 things. You're also going to need some markers, and you need a good black marker. Any black marker will do. I happen to like Sharpie markers, so I've got one black Sharpie marker right here. And then you'll need a few other colors too. So I'll show you what that's for a little bit later on. Now, another thing that you're going to need are popsicle sticks. And so here, these are jumbo popsicle sticks, or the thicker ones. They're kind of like tongue depressors that, in the doctor, they push your tongue down. If you can't get these, that's okay. If you only have the skinnier ones, you could still work with that. We can still make a model with that. We want to use 9 of them, so you want to make sure you have 9. I'm going to make sure I have 2, 4, 6, 8, 9. I've got 9 right there. And then, very important, you're going to need some tape. And so masking tape is pretty much the best tape to use in this situation. But for some reason, if you can't find any masking tape, you can experiment with other types of tape. So it's all kind of like a fun experiment, the whole thing, in the end. And so these are the things that we're going to build our model, and I think it's going to come out pretty cool by the time we're done with it. But first, we have to talk about this. We need to know what this is. Now, some of you, especially if you've ever been to the DNA Learning Center, you might have some idea what this is called. And so if you do know what it's called, just give it in the chat. Throw it in the chat there. I'll take a look and see if you guys, can recognize it. But anyway, so we call this DNA. So we're going to be talking about DNA. We're going to be talking about all the things that make up DNA. So that's structure of DNA. Now, to bring this to you, though, I'm going to share my screen and talk a little bit, write a few things. So let's see if I can, there we go. Screen. This just takes a few seconds for it to kick in. And then I will go over here. Okay, so now deoxyribonucleic acid. So that's a big, long word. And so you can see that there are some letters in red here, right here, D, N, and A. Oh, well, that makes sense. And so that's the full name of DNA, deoxyribonucleic acid. Now, some of you might think to yourself, why? Why do they do that to us? Why do they make these big words that don't really make sense? But they do. And in the bottom line of all this is that little parts of the word make sense to scientists. And I can break down this word into 3 sections right here. And these 3 sections tell me 3 different facts about DNA. And so now the deoxyribo, we're going to get to that later. So I'm not going to explain that right now. So I'm just going to put later for now. And we'll get to that later. But nucleic, that tells you a location. That tells you where you're going to find that DNA. And if you look at that word, you might have heard of a word that sounds a little bit like that. You might have heard it in school. And of course, if you know any of these things I'm talking about, you could always chat them. I'll take a look, see if you get it right. And so, but this nucleic actually comes from another word called nucleus. And a nucleus, in this case, the nucleus is the center of certain things, and we're talking about the nucleus of a cell. Now, actually, in a few days, we're going to have another session on cells, so you can learn a lot more about cells than what I'm going to tell you today. Now, on the other hand, acid, that's a little bit different. And so this kind of tells you about a property of DNA. And so if you've heard of acids, you've probably heard of acids and bases. And so they're also sort of associated with charges too. And so acids tend to have more of a negative charge than bases. So bases tend to be more positive. So yes, DNA is sort of negatively charged. And I'll explain that a little bit more later on. And so now, let's think about where it is. So if I'm looking for my DNA, that means I need to find it inside a cell. So here's just kind of a roundabout little circle of the cell, not perfectly circled, but that's okay. And so the outer part of the cell is what we call the cell membrane. And so that cell membrane protects everything. When you go inside, you're going to find a bunch of gooey stuff like called cytoplasm, and you're going to hit another membrane. And so this next membrane is the nuclear membrane, or the nucleus. And that is where we find our DNA. So the DNA is in that nucleus. Now, if you have the cell from a human and you take that DNA and you pull it out and you stretch it out, you're going to find out it's actually pretty long, because humans, in each of their cells, we have about 6 feet of DNA. Now, that's pretty impressive. Let's think about that for 1 second. Let's say we have a 6-foot person. Let's make it a little bit of a confused person. And we have a cell. There's your cell right there. In fact, that's probably too big for a cell, really, but I'm limited by technology. And so, how do you fit 6 feet in that tiny little space? Seems like totally impossible. And so, well, you have to consider 2 factors. And so the first factor is that DNA is super skinny. So it's extremely skinny. If you want to get a clue as to about how skinny it is, you'd have to take a single strand of hair and you'd have to split that hair down the middle, probably about 1 million times to get the actual thickness of DNA. So DNA is just, super microscopically thin. Now, the next thing that you need to consider, because even if it's microscopically thin, it's still really long and you have to deal with the length. And so to deal with that, we need to look at the shape of DNA. And so I'm going to sort of come back for a second and we're going to look at that model one more time. So I'm going to stop broadcasting and pop back. A second. There we go. And I should be back. There I am. And so now right here, here's our model. And so we have this cool shape. In fact, it's one of my favorite shapes ever. And so if you've ever seen this shape, you might know what it's called, but it has this twisty shape to it. And so that twisty shape helps out with a lot of different things. And so now, first of all, if it's twisty like that, I can tighten it and you'll see it starts to shrink or become more compact. And so that shape allows it to compact really, really neatly. In fact, it compacts into sort of like little bundles. Now I'm going to go back to my screen so I can tell you a little bit more about how that bundles up. There we go. And it takes like 3 seconds each time. There we go. And so if you think about your DNA, it has this sort of double spiral. So we actually call that, the scientists who discovered it, they decided to call it a helix. And they called it a double helix. And that's because it has 2 sides. And so these 2 sides, and the 2 sides, we call those the backbone. We're going to go through that again later on. Those 2 sides can curl up nice and tight. And so the way it curls up, though, I'll kind of give you a basic. It curls up so much that they start to make these bundles. Now, if you're tuning in and you know what they call these bundles, they look sort of like little X's. You can chat that while I'm drawing it right here. take a look. No, I didn't see anything on there. That's right. So we call these little bundles, we call them chromosomes. And these chromosomes, we don't just magically get them. We get them from certain people. We inherit them from our parents. And so you inherit your chromosomes. You inherit a certain number of chromosomes. That's a very bad N right there. There we go. And so we get a total number of 46. That's a nice even number right there. And so we actually get 23 from mom and 23 from dad. And so that's what makes our nice, neat 46. And that also describes why we look a little bit like our parents, why our traits resemble our parents. And that, we'll talk about what DNA is for in a little bit. Now, the whole reason why that DNA can bundle up like that has to do with its shape, that double helix shape. And so therefore, the discovery of the shape of DNA was really, really important. It was discovered in 1953. And I'm going to give us a really brief history lesson of that. And so in 1953, there was a very, very fascinating picture taken of DNA. And so it was taken by this scientist right here, Rosalind Franklin. And so it was actually an X-ray picture of DNA. So they used X-rays. I'll say there we go, of DNA. Right there. And it was taken by Rosalind Franklin right there. And they used to take these X-ray pictures. And she was trying to figure out, like, what is this molecule like? Like, what is this shape really for? Now, her boss decided that he, wanted to know a little bit sooner. And he decided to share this information. So his name is Morris Wilkins. And so he took her pictures and brought them to these two scientists over here. And right here, this is James Watson, and this is Dr. Francis Crick. And Dr. Crick actually had some experience with things that were helical or twisty. And he looked at that X-ray and he kind of identified the shape pretty quickly after that. And so they realized that they were dealing with a double helix. And Watson and Crick were actually the first two people to build a three-dimensional model of DNA. So they got that DNA up really quickly. Now, for this, there was a Nobel Prize. The Nobel Prize was given for this discovery in 1962. Now I'm going to point out who actually got this prize. So Dr. Watson got it, Dr. Crick, and Dr. Wilkins. So what happened over here? What's going on? What happened to Rosalind Franklin? And so, a lot of people said, well, it's because she's a woman. Well, unfortunately, we won't really ever know that because she died in 1958. And so she actually died from cancer. And it was related to the work that she was doing with X-rays. Later on, we figured out that X-rays actually can cause cancer. And so she died very sadly. And when you die, you cannot receive a Nobel Prize because that money is given out to lead the person into new research. And so that's kind of an important point. Now, anyway, so now that we know the history, we want to get down to, well, what is this? What is the function of DNA? What's its purpose? I mean, there's so much of it inside our cell. We need to know what it's all about. And so, if you think about it, like, let's say here's my DNA coming from a cell. So I'm going to try to make it sort of like a double helix like that. And so I consider all of the DNA to be sort of like a cookbook. And when I say cookbook, I mean a cookbook for a living thing. So all living things have a cookbook. The cookbooks aren't all the same. You've got different recipes and things like that, but you have a cookbook. Now, in every cookbook, it's not like you're going to, open a cookbook, start at page 1 and just go. You're not going to make everything in a cookbook. You're going to find out your kitchen is a big mess by the time you're done. And you probably don't have all the stuff to do that. So you look for specific recipes. So you're looking for recipes. And in DNA, we call those recipes genes. And so if you want to think of it this way, those genes are like little sections on your DNA cookbook. So they're like little sections. Now those genes are recipes to build things. And so actually your genes are recipes to build these things called proteins, and they can build or control proteins. And so, genes have a couple different roles here and there. And so basically they're just revolving around making these proteins and making these proteins work. And those proteins, I consider those proteins to be sort of like the building blocks to your traits and your characteristics. And so that's the purpose of DNA. So it has such an important role in living things. Like without this, you wouldn't be able to build a living thing. So you wouldn't have the instructions to do that. So now I want to know, well, what are those instructions made of? Let's get to the tiny parts of DNA. So I want to get to know, like, what is the actual structure? What makes up this cookbook? And so, well, the cookbook, so now think about it. We're thinking of the tiniest units of a living thing. So like, let's think living things. Let's think person first. So we've got a person right here. I'll make it not a confused person. I'll make it a happy person with a weird sideways smile. And so this little person is made up of units. And so the units of basically people, animals, and things like that, we consider the, living things, those units are cells. So living things are made up of cells. But when we're talking about DNA, we need to think smaller. So we need to think of the tiniest things that can make up something like DNA. And it's really the tiniest thing in our universe. And it makes up everything in our universe. So all things are made from these tiny little things. And so the basic parts of DNA, we call them atoms. And so, ultimately, we're made of atoms too. But DNA, the actual structure, is made of very specific types of atoms. Now, atoms, we have a way of categorizing them. And so if you look at this chart right here, the periodic table of the elements, you might have heard of it. Now, when you see the word element right here, we're talking about types of atoms. So we're referring to types of atoms. So these are all the different types of atoms, all different types. And I'm not going to go into how the periodic table works today, because that's a whole other course in itself. So that's going to be probably for another day if you guys are interested in it. But I am going to talk about very specific atoms on this chart, specifically the ones that you find in DNA. So I'm going to circle these little guys. So right here, here's one, H, and I'll go over what they are in a second. And you'll notice, here's C, there's some C in there, there's some N, O, oh wow, these are so close together too. And so that means that they have things in common. They're the same color, they're close together except for H. That one's kind of like a little bit of an outlier, but they're very, very similar. Now I'm going to take those, I'm going to talk about them individually. So right here, you have PONCH. That's how I like to remember the things that are in DNA. Ponch, those are your atoms, like poncho without the O. And so let's go over what they are though. So right here, you have H. And H, that stands for hydrogen. And if you know any of these, you could pop these on the chat. If you want to pop them on there, that's fine. There's hydrogen. And then we have, I'll go to another very common one. O, probably know what this one is. It is oxygen. And then we have C. This one's a little tricky. A lot of people get this one wrong because they automatically jump to carbon dioxide, but it's actually just carbon. Dioxide would be a little too much. We'll talk about that though. And then we have the N, and that is nitrogen. So you might notice, some of these are kind of similar right there. And so yeah, those are actually all types of gases. So they have that in common with each other. And of course, last but not least is that P, which actually doesn't sound like a P. It has more of an F sound, and that is phosphorus. And so it makes up your DNA: phosphorus, oxygen, nitrogen, carbon, and hydrogen. So those are the 5 different atoms in your DNA. And so now, talking about atoms though, they don't like to be hanging out by themselves. And so a lot of the atoms, they like to hang out with other atoms, and it depends on how much space they have. Like if you want to get technical in chemistry, it has to do their electrons, the number of their electrons. And so atoms like to stick together. And when they do that, they form molecules. And so basically a molecule or molecules are 2 or more atoms joined by a bond. There we go. And so they're joined by a bond. And so I'm going to give you some examples. I can actually take them right out of plants right there. So here's one very famous one. You might know what this is, H2O. So now this is, if you know, throw it on that chat. And so this is the chemical formula. So this is a chemical formula right here, the chemical formula for water. Now, a chemical formula just tells you exactly how many atoms are in that molecule and how they fit together. Now, when you don't see any number right there, that means it's 1, because it can't be 0, because then it wouldn't be there. And so right here, so this means H2O means that you have 2 H's, and they're sticking to 1 O, like that. And so now, and if you think about atoms, H's, they can only connect to one thing. They've got only one free arm. Imagine if you only had one arm, you can only hold one person's hand, whereas we have 2 arms, we can hold 2 people's hands. And actually, oxygen is like that. So oxygen has 2 arms and can hold on to 2 hydrogens in that example. And so water, there's a little molecule of water right there. Now I can make another one over here, and this one, CO2. And so now that is the chemical formula. And so you've got 1 C connected to 2 O's. There we go. So that's what the molecule looks like. Now what this stands for is carbon dioxide. So I mentioned that before. And that is the actual chemical formula. Now you might see right here, the oxide part, that's your O. And here's your C, that's pretty obvious. But then this doesn't seem to be represented in there. And that's because that is a number. And so that's the number 2. And you can see, because it's connected to this word right here, it's telling you that there are 2 oxygens and that they stick to carbon. And so that's just how you describe them. So we've got 2 kind of molecules right here. But these are very simple molecules. They're very simple. They are only made from 2 different types of atoms, each of them, even though they're a total of 3 atoms. And so now if you think about DNA is a lot bigger. It's got a lot more parts that come together. And so we call DNA the macromolecule. And so a macromolecule, I kind of love, I love that word. Macro right here means big, right there. And molecule, well, a big molecule. And so the way to define what that means, though, it's basically a molecule made up of molecules. So a molecule made up of molecules. So it means that DNA is made of several molecules. And so now let's go back. I'm going to kind of show you our DNA model one more time. I'm going to stop my broadcast. Going to pop back on. Here we go. I'm back. Great. And so I'm going to look at this little DNA model right here. And so if I look at it, so we have the outside part, and I kind of briefly mentioned that we have the outside part, which is what we call the backbone. And so that's the backbone of your DNA right there. And in the center, I can straighten it out. It looks like a ladder. And so that ladder, here we have the steps of the ladder. So here are those steps. And so we have steps and we have the backbone. So the part that we're going to talk about first, I'm going to talk about that backbone. We're going to talk about this white, black, these alternating colors right here. So we're going to talk about that. And we're going to define what they are and sort of why they make up the backbone. And so I'm going to go back to my screen, just jumping back and forth here. Screen, broadcast, 3, 2, 1, boom. There we go. And go back here. And so now the backbone, let me smooth this up a little bit. Here we go. Oops. Hold on, a little technical difficulty here, but I'm getting there. Here we go. And so we've got 2 parts. And you see, I have the actual chemical shapes for them. And so the white part. So this right here is showing you the white part of DNA. And that little molecule you're seeing there, it's kind of actually a big molecule. That's a type of sugar. Now, the type of sugar that's in your DNA, very interesting name. It's called deoxyribose. Huh, you might have seen that before. And so, yes, that is indeed the D in DNA. And so that is the name of a sugar. OSE tells you that it's a type of sugar right there. And so if you're looking at words like glucose, fructose, sucrose, all of that, those are all types of sugars. Now, if you look at the molecule over here, then you'll see that in it, literally see, there's a lot of C's in it. So there's a lot of C's in that molecule. You'll also see some O's hanging out there and lots of H's. And so that's what makes up sugars. You've got carbon, you've got hydrogen, and you've got oxygen right there. Now I want to point out, remember, this is what's inside your DNA, PONCH. So, well, we've just, we've collected some carbon, some hydrogen, and some oxygen. So we didn't get to the phosphorus or the nitrogen yet, but we will. And so now, so that is the white part. So now that white part, these white parts are connected by these black sections in between them. And so the black sections, so here's the black section right here. And I like to kind of consider them the glue. And so they kind of glue those sugars together, and they are the phosphates. And so that's a phosphate right there. And so you can imagine, well, phosphate, ooh, that tells you there's some phosphorus. Ooh, there's our phosphorus right there. And ATE in chemistry, and you'll learn this in high school, you don't really have to know this now, but that tells you there happens to be 4 oxygens. And you can see here are your 4 oxygens right there. And so the whole molecule is actually what we call PO4. And so that's the whole molecule right there. And that's the glue. So that's what actually glues the sugars together. So it's in between those sugars. And so that's your backbone. And they link together very strongly. So the backbones are strong in DNA. But now we're going to talk about the rungs of DNA. So we're going to talk about the parts that make up the steps. And so if we go here, so molecules of DNA have steps. And these steps, there's 4 different colors. And that's because there are 4 types. And these types of molecules that make up the steps of DNA, we call those nitrogenous bases. And so we're looking at the 4 different types of nitrogenous bases. Adenine, we like to refer to them by their first letter. So A for adenine, T for thymine, G for guanine, and C for cytosine. And so those make up the steps in our DNA. Now I'm going to come out and take a look at our molecule again. So I want to talk about a few things with the model in mind. So here we go. And so here's our model. And so here's your backbone. There's your sugar, your phosphate, sugar, phosphate, sugar, phosphate. It's kind of like SP, SP, SP all the way down. And then the steps in the middle, right here, you'll notice these steps are never by themselves. They like to hang out together. They love to hang out together. And so they have a certain way that they hang out together. And if you take a look at my model, so you can see, you'll see that the colors are very specific. Here you have your red, and this is a yellow over here. Here you have a blue, and that's a green. And you never see that blue hanging out with anyone else. You never see a green hanging out with anyone else. And so, and the same thing for the red and the yellow. So they pair up. They like to pair up very specifically. And so the way they like to pair up is A loves T, and then G loves C. That's how they like to pair up. So A with T and G with C. And so they link together. That means that you can have A and T, you can have T and A, either way, doesn't matter, but you can't have A and G, or T and C, or T and G, or anything like that. So they have to kind of stick with the pair that they want to be with. So sort of like linked for life, so to speak. And so now let me just go back to my diagram again. Gotta wait for that little, there we go. Right there. And so now, so they're linked, and it's very interesting because they're linked by little hydrogens. And the number of hydrogens between them, there's, like it's not just one hydrogen that links them together. So between A and T, there are two hydrogens. And so if you want to think of them as little hearts, like two hearts, how cute is that? There you go. And now between G and C, there are three hydrogens. So there's three little hearts. There we go. If you're into the romantic idea of it. And so these hearts link these two bases together. So these bases, and by the way, the nitrogen, that's within each of these bases. And so in nitrogenous bases, you have nitrogen, you have hydrogen, and you have oxygen. And bases are basically like little combinations of O's and H's that kind of combine like that. And so if you want to remember how many of these little H's are between them, if you want to think that G and C have a greater love, that's up to you. That's fine. That just helps me out remembering which one has three and which one has two. And so there you have it, and they come together. Now, they're not always like that. And so, when you're trying to create DNA, or rather when you're trying to take DNA and make more of DNA, then you have bits and pieces in the nucleus. And so these bits and pieces are ready to come together. And we call these nucleotides. And so a nucleotide is a base. So right here's the A base connected to a sugar, that's your sugar, and connected to a phosphate. So inside your nucleus, you can see I kind of outlined it right here. You have all these little units right here. And so when your DNA is ready to make more, those units will come in and fill the spaces. I'll describe this a little more later. So that's just something, kind of to prepare you. But anyway, when we put all of this together, our DNA molecule actually looks like this. And so right here, like here's your phosphate, right there, there's your sugar. So you have your sugar phosphate backbone right there. And then in the middle, you have your bases, but they're paired up. So that's why we call them base pairs. So they're base pairs, they're paired up. Now, if you want to know how many base pairs we have as humans, we have 3.2 billion. Yep, 3.2 billion base pairs. We have a lot of base pairs, and that makes up that whole 6 feet of DNA, so 3.2 billion. Now, in that 3.2 billion, in those 3.2 billion base pairs, we have approximately, I'm just going to go down the middle number, we have about 27,000 genes. So think about it, 27,000 recipes inside that DNA. So within that little code that makes us up. So we have this, and that's the genetic code right there, our A's, T's, G's, and C's. So now we've learned a lot about this. So now we're going to build a model, and I'm going to use this sort of like almost like a template. So that means I'm going to get out of here and I'm going to come back to the screen where you can actually see me. There we go. And so now I'm going to go back to my spot over here. Okay, so now we've got all these things, and I am going to, first thing I need is tape. And so you want to make this tape, and this is good idea to kind of, get a good idea of how long you need your piece of tape. I want my piece of tape to be longer than my pipe cleaner. That's kind of important. So I'm just going to lay that down just so I've got a basic idea. Now, I'm going to take my piece of tape. There we go. Make sure that it's longer. I'm going to rip it off. There we go. Now, here's where a lot of people mess up sometimes, because you might think, oh, let me stick to the table. No, you don't want to stick to the table, but you do want it to stick to the table. But you don't want the sticky side down, because then you won't be able to build a model that way. And so what we're going to do is I'm going to pinch the end. So there's only a little sticky part facing down, just a little bit on each end. And then I'm going to stick those down to the table with the sticky side facing up. So if I do this, like this, kind of scoop my thumbs off of it, now it's sticking to the table, but most of the sticky side is up. So that's good. Now I want to do the same thing for the other side, but you don't want to make them too close and you don't want to make them too far away. And so I'm going to use my popsicle stick as a judge, because you want your popsicle stick to be able to reach across. So really, you want to make sure the two pieces of tape are about 2 to 3 inches apart from each other. But you could use the popsicle stick as a guide. Okay, it can be, you know, well, you want it to be about the same size. There we go. And I'm going to make them about, that's a little long, but that's okay. I'm going to make it about 3 inches apart. There we go. There we go. And I double check because I can see that my popsicle stick will actually fit across there. So that's good. Now come the pipe cleaners. So the pipe cleaners, they're going to give some support to my model. That's why, you know, the wires are important. Not totally essential. Technically, if your model doesn't have support, it's not the end of the world, but it's definitely more fun with these guys. So I'm going to put this in the center of the tape. And I would squish it down a little bit. Don't touch the tape because then it gets stuck on your fingers, but just squish your little pipe cleaner down a bit. There we go. And now I'm going to try to even them out a little bit. Stuck on there. Oh, well, that's it. Squish that one down. There we go. Okay. So now that's good. Okay. I'm not even quite halfway there yet. So now I need my jumbo popsicle sticks. These are going to act as our base pairs. And so we're going to lay them out. I definitely like to put one over the edges of the pipe cleaners because later I don't want that sticking up and annoying me by poking me in my finger. And then I'm just going to spread these out because the pipe cleaners are, you know, they give you some room away from the tape, so it's not going to stick. You can kind of even them out a little bit, make them look nice. Doing good. There we go. And so now I'm going to squish them in place. So that means I need more tape. This is another danger zone for people because sometimes if you're not that good with tape, this is where it all goes crazy. You need to a lot of people make the mistake and they're like, yeah, I got this. And now, wow, I've seen that backfire a lot of times. You're going to take a piece of tape, you know, you want it to be around, it can be longer, that's okay. And you're going to rip it off because this is going to make it so much easier. Then you're going to take that, this time it's sticky side down, but you can sandwich all of that stuff inside. And you're going to take that piece of tape and you're going to hover it above the other piece of tape with the popsicle sticks and the pipe cleaners in between. And then you're just going to go nice and close and whoo, there we go. It's okay if it's not perfect. Believe me, I actually missed a little. So now you're going to squish it down. So I'm going to squish it down really good. There we go. Make sure, get in between there, because pipe cleaners, they're kind of thick, so you got to make sure that it sticks to them. Now, I'm going to do the other side. So same thing. There we go. I'm going to sandwich that in. And my model's not really perfect. Okay, got a little crooked, but that's okay. And so now, you might be thinking to yourself, wow, she just made train tracks. And so, yeah, well, we're going to turn these into something better than just train tracks. So now first, I want to clean this up. So now, I can just rip it. If you have scissors, you can trim them, that's fine. Whatever you like to do is okay by me. In fact, I got my pipe cleaner stuck out a little bit, so I'm going to bend it over. There we go. Going to trim the other side. There we go. Garbage. And now I've got my trimmed model. So now this is meaningless, but I'm going to squish down my tape unless I start to label it. So now I want to label the parts. So in the resources, if you did print some stuff out, you might see that we've got some diagrams, and this is just meant as a guide. And so this just shows you sort of how it all comes together. And so you have your sugars and your phosphates. There's your backbone. Here's your base pairs. Now these base pairs, it doesn't have to be in this order. You can have any base pairs you want. And so you could have it all AT if you want. So we're all GC, whichever. And so you can make up the sequence. And so for this, first I'm going to label my backbone. And so I'm going to take my Sharpie marker. And so we have, now if you look at this, here's your little base pair. Now let me look at my model for a second. And so here we have our base pairs. And if you notice right here, you'll notice that those base pairs are only connecting to one color. They're not connecting to both. So they're only connecting to the white. They're only connecting to the sugars. Basically, the big reason why is because phosphate only has enough arms to connect to two sugars, and that's it. So it doesn't have enough room to go and connect to something else. And so therefore the sugars have enough room to grab onto these arms right. And so when we make our model, if you look, so right here, this is where the sugars are going to be. They're going to be where the tape is on your popsicle sticks. Now in between, where you see the little pipe cleaners sticking out, those are going to be where your phosphates are. So think pipe cleaners, that's where your P's are going to go. And so I'm just going to write S's really quick. And so now I'm going to do the other side. So the way you kind of think of this, I mean, the way I like to remember it is SP, 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 or like salt and pepper. So salt and pepper, salt and pepper, salt and pepper. There we go. And for your model, you could think P for pipe cleaner, because that's where you can see your pipe cleaner through the tape. Oh, that's a little P right there. There we go. So now I've just labeled my backbone. And so you have your S's and your little P's are gluing them all together. Okay, so that means I need to come up with a DNA sequence right here. And so for that, I'm going to just make any sequence I want. And so I'm going to do, you know what? I'm going to do cat tag tack. So I'll show you what I'm talking about. I'm going to write my sequence on cat tag tack. There we go. So if you look at that, cat tag tack. So only one side. So I came up with my own sequence of DNA on one side. Now I have to figure out, okay, well, who's going to pair up with that? So with the C, what's going to go with your C? And so with your C, remember greater love, that's going to connect with your G. So there's your G right there. A is going to connect with your T right there. I drew a very bad T. T is going to go with A. T, A, oops, T, see, even I mess up. And then G and C, T and A. It's hard for me to draw this way, but that's okay. There we go. So I've kind of filled it in. And by the way, if you do mess up and you're like, I ruined my model. Well, actually you didn't, because you have a whole blank side. So you can always redo it on the other side. But I'm not going to do that. I only kind of just like smushed up my T right there. So now to make it pretty, you can color it. So you're going to pick colors, and you can pick colors for your S's and P's, but I'm going to just pick colors for the letters, the A's, C's, G's, and C's. And so you can pick any colors you want, except, so for example, let's say I'm going to pick G for green. That kind of makes sense to me. Can't explain why. But so you can color. Now, if I color this one G green, am I going to go and color the next G pink? No, that's silly. So I'm going to keep all of my G's the same color. So same thing with all my A's, my T's. So they're all going to be the same color. And so now, but now let's just say, okay, I'll pick blue for my C. There we go. And I'm not going to, spend a lot of time making you watch me color these like, Da Vinci or anything like that. I'm just going to scribble it on there. There we go. And then, you know what? Get my red. And I'll leave T just wood colored. Of course, you guys can make this much prettier than I'm making it, because I don't want to have you guys watching, just like I said before. There we go. Now, also what you can do you can show the little bonds. And so if you wanted to do like, you have your CG, if you wanted to do 3 hearts like that, or 2 hearts, or if you're not into hearts, you're like, no, I'm not into romances. Sorry, no, I don't like that. And so then you could just put little H's, but it's a good way to remember how many like little bonds are between there. I'll go to skip down to this little GC. I got my 3 little H's right there. And so there's my little bonds. You can color, I would color them up to there if I was like not, scribbling this out. So now, okay, so now I have this not only fully labeled, I've got it colored. So this is cool. It gives you an idea. It'll kind of make it stick in your head about as far as what parts of DNA there are. But you're like, oh, can I twist it? And like this, you really can't twist it too well like this. But I do have a trick. And so now this is a tricky trick. So I'm going to use the camera to show you. So now here's your DNA model. And so what I like to do, I want to get this to twist just a little bit. So I'm going to grab the top rung like this. And I'm going to take the one underneath it. And I'm going to bend it behind and over, just like that. And now I'm going to do that with this one too. And I'm going to keep bending them back and behind. You might notice it starts to look like a fan. So I'm going to keep going. And if you're always bending it back, then you're going to, be doing, oh, this one's a little tight, but that's okay. There we go. And so now you can see, looks like a fan. There we go. But now I'm going to straighten it out a little bit. And now your DNA model has a bit of a twist, a little bit of a twist. So now, of course, when you go to do this, you might have more than 9 of these popsicle sticks. So you might want to make it longer. So I did a longer one just to give you an idea of what this looks like. And so right here, I decided to make a nice long one here. And so this one looks pretty cool. It's got a cool little twist to it. And so I think this is about 18 sticks like that. And so, same thing, you just keep going, keep twisting them, and then it's got this kind of little cool twist to it right there. Now, just to show you, there are other ways to make models too. I decided to, just give it a shot. And I actually was able to make them out of pipe cleaners. It was a devotion though. I was devoted to doing this. So if you are that person who really does want to do it, this will work. It did take me some time and it was a little tricky. So I will say that, but it's a challenge. So if you like challenges, you can kind of twist it if you're gentle. And then that's another way that you can get kind of a DNA model out of it. And of course, if you like this model, I'll just use this. I think I put mine somewhere else. So this little model right here. And so we do have pieces to build this. You can buy this with a little kit, and you can find that in the resources. So if you're interested in buying the kit and building that little model, having that in your little collection. And so now, and that about, except for one more thing, I just want to show you how DNA replicates or copies itself. So now, if you have this DNA right here, and you need to make a new cell, you need to make this into a new cell. So the way you're going to do it, you're going to need to open it up. You're going to need to unzip that DNA. So here I have this little model, it's kind of a little bit unzipped right here. So that means you're going to pull apart the hydrogens like that. And so you can unzip it. Technically, the whole thing would get unzipped, but doesn't get unzipped all at once. And then when you're in the nucleus, your nucleus, again, is full of those little nucleotides. So when your DNA is ready to make new pieces, those new pieces are going to come in and attach to each side of your DNA. And so they're going to come in and attach like this. I can attach these like this. I can't really attach the phosphates very easily with my model, but you can see how that's starting to build the other side of DNA. And eventually these are all kind of like stacked up like that. And both of these models would split with both sides complete, and you'd have 2 strands of DNA. And so now, I told you lots and lots of stuff about DNA. And so in one of our next segments, you'll hear lots and lots of stuff about cells and things like that. And so I hope you enjoyed our session of DNALC Live. and I will see you next time. Have a great day.